Can you move the slide, please? It's my pleasure to introduce the next speaker. The next speaker is Stefano PICCOLO. Stefano PICCOLO from Italy, from Padua, from the FIRC Institute, Institute of Molecular Oncology, and in Padua School of Medicine. And Stefano did a lot of seminal work about mechanotransduction and how it impacts on tumor heterogeneity, oncogenicity, and plasticity, and how it can maintain the different cell state within a tumor. And we are really happy to have Stefano here today. And don't hesitate to ask some question on the field of the chat. Thank you, Stefano. Welcome.
Thank you. Can you hear me?
Yes.
And do you see my screen?
Yes, perfect.
Excellent. So thank you so much for this invitation. Thanks, Danielle. Thanks to the organizers. So what we are interested in is our emergent properties, that the properties of communities where wonderful things happens when different cells lives into a complex ecosystem and start gaining abilities that are absolutely unique and proper of that layer of complexity. And you just heard right from Hans, self-organization in organoids, in iPS cells, even in cells that I've never seen, a body building from, initially from Yoshiki's eye, neural structures, eyes, and pieces of brains, but also, very classic events of contact inhibition that we see every day in our tissue cultures, and of course, development. All these are examples of social behaviors with unique properties that are not recapitulated by individual cells. And this happens all the time when cells are perceiving each other in a sense outside of what you may think the dictatorship of plastic, of petri dishes. You need to let them, if you want, express themselves, unfolds inner scripts that can, build self-morphogenesis. So I apologize. Okay, so there are beautiful examples of these social behaviors, and these things happens when cells start touching each other in communities. So for years, people have been fascinated by the fact that cells can change their behavior when they start feeling each other physically. And the attachment of the matrix, of course, is very important, and so forth. We know that there are integrins, there are special channels on the cell membrane, like, famous Nobel Prize of this year, piezo. There are, of course, integrins and cellular matrix, and so forth. This is organized in skeleton, but at the end of the day, we know that it is changing gene expression and it's changing cell fate. So how to couple all this to this physical environment, to gene expression, has been for years a mystery. And we contributed to this with the discovery of YAPAs as factors of this pathway called the HIPPO pathway, nuclear transcriptional regulators as in fact mediators of mechanotransduction. This is work, seminal work done by DuPont et al. And then Aragona some times ago. Now, since then, YAPAs mechanotransduction has been a global evolutionary conserved sensor of different mechanical cues in different contexts and different cell types, going well beyond, our original findings using individual cells in stiff and soft, big and small adhesive areas, and so forth. But also in 3D cultures, similar to the organoids that you have been discussing, work, for instance, by Matthias Ludolf very interesting in this respect, but also responds to pressure, to disturb blood flow in atherosclerosis, and so forth. So what we have been particularly interested in is the function of mechanotransduction as seen through YAPTAZ in vivo. So think about plasticity, intended here as the capacity to reversibly change transcriptional profiles, shape, fate, and potency of cells. So I would like to tell you a bit about our findings on YAPTAZ in adult cellular lineages. So case number one is the activity of YAPTAZ required to convert normal cells into their corresponding tissue-specific stem cells, normal stem cells. So we started this with a mammary gland, although we extended this notion to additional paradigms. So this started from work by Tito Pancera, where he was considering that endogenous YAP was required, based on our prior work, for keep organoid expanding, ex vivo generation and preservation of somatic stem cells, we would say, and we said at that time. So we reasoned, what about if we do, Yamanaka type of experiment, in which you overexpress YAP or TAZ in a inducible manner in differentiated cells. So what we did is to FIRC sort luminal differentiated cells from, Cedric mice, just to make sure that these were luminal cells using K8 GFP drivers, and infect these cells with RTTA and eteton YAP, and then just wait at doxycycline. And quite amazingly, although control cells remain as single individual cells, you started to see mammosphere popping up in the culture medium. That after this mammosphere, you can culture, self-renew, and even push them to build a mammary organoid. And so we asked what happens to these cells. What are these cells? So by transcriptomic analysis, actually the sequencing and the computer could not discriminate between these YAP-induced organoid, which we call Y masks, from basal cells that use no ex vivo regain capacity of mammary stem cells. Now the two are indistinguishable, and injection of these induced stem cells that were formerly luminal cells was able to regenerate the mammary tree and to differentiate in the different three histological lineages of the mammary gland. So this is not the only story, which is about normal one, but about transformation. Now, there is a huge interest of YAPTAZ in the cancer field. We reviewed this a few times. And there are basically no tumor in which showing upregulation of YAPTAZ in solid tumors, although there are some exceptions, like non-small cell lung cancer, for instance, small cell lung cancer, in which there is no relevance of YAP. But in general, YAP is actually massively upregulated in most human tumors and in mouse tumors. So our work during years, and this is just a selection, this is by no means the whole thing. Many other groups have contributed to that. This is just what we did. Contributed to the view that YAPTAZ are critical for steps in tumorigenesis. These are all experiments done at the tumor initiation level, I need to stress, in the context of intestine, liver cancer, skin cancer, and pancreatic cancer. Now, this is, of course, highlighting the relevance at the functional level, not just association, in pathological samples. And this has initiated a large interest in the pharmacological industry in developing a form of YAP-TAZ inhibitor. These are very, very hard to tackle, but there are tools. You heard some of them, like from Cedric Lampain, from SARC inhibitors. Of course, SARC because it essentially fits on mechanotransduction. It is true that asatinib, it can work, but it can be toxic. But there are new SARC inhibitors that can be used, and are ROCK inhibitors, and also factors working in the nucleus, like BET inhibitors, that are critical for YAP-TAZ to operate on their target genes, that are mainly super-enhancer type of cis elements in the genome, something that I will have no time to go in. So truly the story is started in this cancer arena a long time ago with Michelangelo Cordenozzi, who was using cell lines at that time, MCF10A type of cells, transformed with RAS. And he was playing with gain and loss of TAZ. And clearly, if you reduce TAZ, you have less mammosphere, less tumorigenic potential in vivo, and the opposite if you boost it. So clearly, the background in which we would like to see this, and would like always to bring your attention, is that cancer is of course a corruption of cell and tissue, social behavior, but cancer is going well beyond mutations. This is a nice review a long time ago by Bissell and Hines, who asked, why don't we get more cancer? And the reason is that, of course, the microenvironment is key, and aberrant tissue architecture is known to promote tumor aggressiveness. And there are, some fantastic examples, and this is just my selection, but there are more, in which the normal microenvironment, if you think, is the most potent tumor suppressor. Because this is an emergent property, something I was alluding before, in which tumor cells are perceived by neighborhood and typically eliminated, as nicely shown by this paper from Phil Jones here on the right. And the need that you need a supracellular rules of engagement is also clear by sequencing efforts, in which if you look at closer inspection, the mutation that you find in normal aged tissues are not that dissimilar from those that you find in a tumor, although these are by all means absolutely normal tissues. That is tell you that the tissue is completely able to keep control, if not to eliminate tumor cells. And we believe that mechanics is key for these events. Now, again, I need to give credit here to Van Weaver, who has put forward, in a sense, this notion several years ago, using experiments on different rigidities and mammary acini, and just increasing stiffness would induce these cells to behave badly, okay? And mimicking, if you want, aggressiveness. Now the question is, if mechanics is important, how is mechanic and genetics holding together? How did they talk to each other? And so here I would like to tell you a little bit of a story about YAP-TAZ as mediators, tyrosine kinase, and RAS transformation. And the end point of this, where I'm going, is that mechanics is, of course, controlling YAP, but that oncogenes of the RTK RAC pathway through the cytoskeleton, are feeding on YAP, and YAP mediates the effect of RTK RAS signaling. So the idea is that the physicality of why don't we get more cancer, right? So because the physicality of the environment matters for oncogene function. And again, I would like to tell you an experiment that was very similar to that one I presented you with YAP or TAZ, just done with one of the most potent oncogene, which is HER2. Basically, same thing. If you overexpress in terminally differentiated luminal cells HER2, which is the cell of origin, of course, of luminal cells are the cell of origin of all type of breast cancer. And again, you see in colony medium colonies coming up, and then you can actually develop organoids out of these colonies. And these cells, however, do behave in this way after oncogenic addition, simply because you do the experiment on tissue culture plastic. If you now go back to the normal stiffness of a normal mammary gland, even if you overexpress HER2, there is no transformation. So that means that the oncogene plus sufficient mechanical signal needs, it's a recipe, if you want, to convert a normal cells into a tumorigenic one in vivo. And second, then if we blunt cell mechanics, we should remove one of these two factors and prevent cancer initiation. Now, to do this experiment, again, we rely on work of other people, like inspired by the designer matrices of Matthias Rudolph and previous work by Val Weaver. And what they have done, is to develop designer matrices in which you can tune stiffness and measure, respectively, the difference in stiffness between normal and tumors. And you can see there are ranges that are not amazingly different. You go from 0.5 to 1, 2, 3, perhaps, kilopascal. So it's not tenfold, okay? Anyway. So what we have done is engineering tumor initiation in human cells. So we took HER2 cells, put them back in vivo, but in the context of a very soft environment that approximates that one of the normal mammary gland. And what you get is basically nothing, is a tiny duct. However, if you now increase in these designer matrices only the stiffness parameter, what you get is growth, an aggressive type of growth, and even seeding HER2 positive cells as dormant cells in the lung. So this tells you that physicality against matter a lot in vivo, and oncogenes need to cooperate with something happens on the outside of the cells and on the ability of cells to perceive mechanics. This is by no means the only experiment. We have seen similar things in pancreas, ex vivo, using this pancreatic organoids culture, starting from acinar cells, this first transition that you have from a normal cells to a panin, it's called acinar to ductal metaplasia, or ADM. ADM is dependent on two things. You need to have an oncogene, but you need to find sufficient mechanics to pull against. To the extent that if you now kill mechanics through, for instance, fasudil, which is a ROCK inhibitor, you actually prevent in a mouse KRAS model emergence of pancreatic cancer. So then we can ask this type of question. Then how can an oncogene ever lead to a tumor outgrowth if the starting cells is a normal cells in a soft tissue? Why does it typically fail? And I believe it fails a lot. Well, you need to consider that there are two elements. That certainly there is a sufficient pulling capacity against the substrate, but at the same time, oncogene feeds on the same pathway. Oncogene change cell shape. Oncogene induces changes in the cytoskeleton and actually change material properties of the cells themselves. It's very easy. You can place a RAS transform cells into and carry out the gel contraction assay, and which are on the left here, very clearly, tumor cells pulse like crazy. So what's going on? So if you don't have sufficient stiffness, as in 0.5 kilopascal, there is no hope for oncogenes to transform a normal cell. There is no hope to send YAPTAZ to the nucleus. However, differently from normal cells, a RAS transform, HER2 transform cells can extract a lot of mechanical information as soon as send yaptads to the nucleus and initiate transformation as soon as, you know, the value approach that one are raised compared to control cells and approach those of tumors. Now, a third example of plasticity and in connection to yaptads is back to normal cells in the context of tissue repair. You know, this is famous statement by Hal Dvorak, who wrote that tumors are wounds that never heal. There is, of course, an intimate connection between tumorigenesis and repair, and this is truly epitomized by the ability of yaptads. So yaptads are largely dispensable for normal tissue during homeostasis. But the take-home message is that yaptads are master genes of regeneration and cancer and organoid ex vivo, setting, you know, a major difference, at least in epithelia, between the rules of engagement that you find under homeostasis and those that operate during a shared program, yaptads ruled of regeneration cancer. Now, for example, a classic example of regrowth is the mammary gland. You know, after lactation, you have involution and regeneration of the virgin state. There is no requirement whatsoever of yaptads up to lactation, but however, yaptads are key for the development of a new secondary mammary gland after involution. Now, this was, you know, something that we originally found in the intestine, so people were very shocked at the beginning to find no relevance of yaptads so in the intestine. However, this is a collaboration I need to stress with Kim Jensen at BRIC, you know, who has led the study on intestinal regeneration. If you give an inflammation like DSS, viral infection, whatever, of course there is a moment of crisis, there are sickness, but there is a rapid regeneration of the intestine. However, this is not the case when you have a yaptase mutant intestine, although the homeostasis is completely fine. And the reason is, very intriguingly, is that actually what yaptads do is to reprogram cells, similar to what I've been telling you, reprogramming normal cells, regain of stem cells, reprogramming downstream of oncogene, initiation, okay? But in truth, at closer inspection, at least in this context, and we suspect in many other contexts, what yaptads are doing are certainly regenerating, certainly awakening stemness. But this is not the stemness that you are used to think in an adult body. Similarly, the stem cell lineage of the adult are as gone as the differentiated one during tissue repair. Just in those 2 weeks on which you repair the intestine is transiently going back to embryonic, fetal, intestinal fate. So the repair cells are directed transiently, and this is what yaptads are doing in the intestine in this case, into a more primitive state. And more or less the same, you know, happens in the context of regenerating organoid ex vivo that are proxy of tissue repair. So we had discovered this when we started to, you know, investigate the genetics of yaptads. We were surprised to see that there is no way you can split organoids for ex vivo crypt expansion. And then, you know, from there, you know, other people like Prisca Liberali and Matthias Lutov put forward, you know, this idea that in fact, as soon as you split organoids, you repristinate transiently cell states that are very unique. And this is entirely YAP dependent. So essentially you go back to stemness, to a really primitive stemness, I have to say, through this YAP as a time machine. Now, the idea that YAPTAs are key for tumor initiation is interesting. But inducing proliferation is by far not what YAPTAs only do or in general do. And we are starting to look into this closer and closer. And this is the case of GBM, where there is no relevance of YAPTAs for proliferation, but for another form of fate plasticity, which is the equilibrium between stemness and differentiation. Again, let me start with the common knowledge and a beautiful work here by Mario Suba and in Cell a few years ago, where, as you all know, glioblastoma multiforme is a high intertumoral heterogeneity. That's a hallmark of this tumor. And there is, as they described, massive fluctuation between the cell state. It's a very chaotic environment. So that, distinctions between stem and non-stem in glioblastoma stem cells at single cell level has proven to be challenging. And particularly because cells are then typically cultured ex vivo as gliosphere, and so we wanted to know what is the nature of stemness in the primitive glioblastoma stem cells in the brain of the patient. So what we did was to carry out analysis of published single cell data sets to identify transcriptional programs of the native glioblastoma stem cells. So we applied this type of algorithms that allows to reconstruct differentiation pathway, developmental pathways. And here on the left, the red cells seems to be the starting point of this neoplasia. And so we could now infer specific glioblastoma stem cells out of this single cell data, and then go through a pipeline that we call RADOANT for identification of masked transcription regulators, through this network reconstruction, pruning of this interaction, use ATAC-seq to infer master regulators. And what we ended up is with YAPTAs being at the top of the hierarchy, controlling other transcription factors in these native glioblastoma stem cells. And the cool experiment is that you can switch tissue culture media in these primary cells from patients and back and forth from a stem to a differentiated cells. And this change of fate induced by the media requires YAP, because without YAPTAs, you don't go back to a stemness, even if you put cells back to a stem cell media. To the extent that if we now start treating established lesion in vivo and now pull the plug to YAPTAs depletion, what you can see is that you have a wholesale differentiation, okay? So these are primary transformed astrocytes that are similar to the cell of origin of brain tumors. In controls, there is dual GFP-TUJ that are, there are neuronal and lineages and the GFP injected cells. You can see that they are different in the controls, but these cells become back to a neuronal state after YAPTAs depletion. So the view so far is that these are not factor essential for homeostasis. This is the picture so far. This is what is actually making YAPTAs very attractive target in the pharma business, because there is apparently a large window of opportunity in therapy. Unless, of course, there is repair, and they are critical for repair and transformation. Although, please let me stress that there are other attributes of YAPTAZ in the microenvironments that need to be fully defined. And a warning, however, to this picture that we contributed to build comes from the last bit of my seminar, which is carrying out, again, genetics, that's what we do, and mechanism genetics, which is a bit changing our perspective. So this is all nice and tidy if you look at epithelia, but tissue are more complex than that. There are stromal cells, for instance, and this is absolutely relevant for cancer because aging is known to be a field cancerization effect. There is changes as we age, which is the most potent carcinogen, of course, aging. It changed the structure of the matrix. It changed, the disposition of the cells, the architecture. Fibroblasts do change, and the ecosystem is a different one. So what mechanism is underlining these age-dependent changes and their eventual impact on cancer is actually still a mystery. We know that there is inflammation. There is this senescence-associated secretory phenotype, which is essentially a bunch of inflammatory cytokines. But what is behind all this? So what we have been considering is what about YAPTAZ activity during aging, but looking particularly in the stroma. And strikingly, what we found is that there are declining levels of mechanosignaling if you go from top to bottom over aging. Already at mid-age, if you follow the white signal, which is the nuclear YAPTAZ, versus the green cytoplasmic of YAPTAZ, you can clearly see that already at mid-age, these mice, unmanipulated mice, start losing the ability to keep YAPTAZ in the nucleus. And the reason is that the mechanical properties of tissues are changing. And we could monitor this by monitor contractility using phosphomyosin light chain. There is clearly much less contractility as we age. And what I've been looking here is, in fact, in the stroma, in the dermis, and we could look at other connective tissue, and this is the same story. You could look at, for instance, vascular tissue and the effect of loss of contractility. Look at the red signal on the right is dramatic, and it is dramatic, the loss of nuclear white YAPTAZ signal in these pictures. And if you look at signatures of YAPTAZ, you can go back to tabula muris, and the tabula muris shows that YAPTAZ mechanotransduction massively decline in a wealth of connective tissue during aging, whereas epithelial and other cell types seems to age with YAPTAZ independent mechanism if they age at all. So we have been exploring this genetically, knocking out YAPTAZ in the stromal tissues, in connective tissue, using the inducible tamoxifen-dependent collagen 1A2 driver, which is very established. Okay? And the result is that we have, very shortened lifespan, dramatic lifespan. And that actually this tissue, multi-organ, you see, traits of aging, induction of senescence, loss of cells. And quite remarkably, these are similar to the defects you observe in old control mice. And if you now use a pulsating regimen of transgenic YAP mice, and you are, persistent, many months of the experiment, twice a week, low dose of tamoxifen. Very striking. What you can see is that regaining of YAP activity is actually preventing the accumulation of defects of old age. So YAPTAs are key in stromal tissue to preserve usefulness. So this is changing a little bit, the criteria that we have been discussing. Before, there is importance in normal homeostasis of YAPTAs, only not in the epithelia, in other tissues that we have never been looked so far. This is another example, smooth muscle cells. There is no pathology that tell a difference between the YAPTAs smooth muscle knockout and aneurysms compared to old vessels. And to the extent that we can, accelerate these events using a mutant in the extracellular matrix in the elastic fibers. This is a fibrillin mutant, the causative gene of Marfan. And if you overexpress YAP in vivo in this with transgenics, you actually rescue amazingly this old-like phenotype. So this is the last information I wanted to share with you. So I'm privileged to work with an exciting team of people, and also an exciting team of collaborators. So thank you so much for your time, and I hope this was interesting for you. I'm happy to take questions.
So, Stefano, thank you very much for this fantastic talk. There is a couple of questions. You illustrate very well the role of YAPTAs in cancer initiation, and the role of YAPTAs in regeneration. For that, there is one question. What about the role of inflammation and the dialogue between YAPTAs, mechanotransduction, and inflammation? Because in regeneration, inflammation is very important, and probably it increases the stiffness of the tissue. So do you think there is some specific things?
I personally believe a lot in this, and I think that likely part of the effect of inflammation are due to changes in stiffness and in other physical properties, not only fitness, like porosity of the ECM, viscosity, mechanics in general terms, and this is changing not only epithelial cells, but also stromal cells. So I do believe that. And beyond that, what I believe or not, the data from the experiment done together with Kim Jensen on the intestine clearly shows that inhibiting mechanics may inhibit the ability of regeneration, not only when you remove YAP, but when you remove other factors like, for instance, SARC inhibitors and other tools. And so we think that this is potentially an interesting area of investigation.
Okay. There is
Can you see me? Can you see me?
Yes.
Because I see myself very, very badly. Okay, fine.
Okay. There is another question about how does reduced YAP activity in aging correspond to increase cancer risk in age?
We can only speculate in that. We believe that there is, it's complex, right? So the easy answer would be, okay, you raise inflammation, and inflammation is pro-tumorigenic, okay? I think that's the easiest possibility, although I think there may be more than that, depending on the tumor stage, let's put it this way. Because at the same time, inflammation is important for not only innate, but also adaptive immunity. It's a matter of dosage. Then too much is going to exhaust T cells. It's oversimplification here can be damaging. So I think we need to make experiments and then discuss about it. I think it's exciting though. Yeah.
Mm-hmm. And there is another question about how mechanotransduction modulates signaling, and is there a direct effect in changing conformation, allosteric effect in particular?
That's the multi-million-dollar question. How is really mechanotransduction works? And I dream to know. I'm not convinced nobody has the answer. Mm-hmm. In mechanistic terms, you mean. So,
Stefano,
it's a if you want.
Daniel, that's me who have the question. I'm also dreaming about that, but I have no answer. You hear me?
Yes.
Yes. There is another question about EMT transcription factor and YAPTAZ. And what do you think about their transcriptional interaction?
Transcriptional interaction? I know what Brablet's presented and no more than that. Mm-hmm. We are not studying this. We are not studying EMT. I mean, we have been studying EMT in the first paper, right? Where we show, but that's the only one, okay? 10 years ago, in the coordinosis cell paper, we show that disturbance of cell adhesion and inducing EMT through twist, snail, and so forth is changing cells and is inducing YAP. And if you knock out YAP, this pro-stemness effect of EMT are gone. So since then, I think we were focusing on YAP and mechanics, and I don't know. I can only speculate that perhaps the EMT state, in addition to all the rest that you heard, you know, these fantastic stories from Cedric, these intermediate stages from Thomas, and what Bob Weimer will tell you later on, are also part of changes in the ability of cells to perceive mechanics, because the cell is changed and the cytoskeleton is changed. So it's a little bit like, you know, raising or lowering the thresholds for YAP activation. I think this all the time you will have a YAP component. I mean, and the FAT1 story from Cedric actually tells you already that more likely this is what is going on. But
there is another question about how much YAP and TAS compensate for the loss of each other in your model, and can you explain how can the variation across models?
Yeah, I frequently have this question, and very pragmatic. I have only a pragmatic perspective that they can cross-regulate each other, so they can make you some tricks when you don't knock them out both. And since they are so overlapping in function, we just, you know, prefer to waste more energies and time in having double knockouts. And whether, you know, YAP is more important than TADS in some context or others, seems to me, in my level of understanding, more of a detail. So we don't study that, but likely I'm wrong. So our stuff, it's only double knockout. But it's for simple-minded perspective, not for logic.
Okay, and now a last question.
I know it's a bad answer, but that's the truth.
About tumor, and specifically glioblastoma. and you talked about YAPTAs and different cell state modeling within the tumor. But how do you explain, for example, in glioblastoma, there is a lot of niches of necrosis area, and in this necrosis area, probably you have softness because this is necrotic, and the people have described that in that niche specifically, you have stem cells. So how do you explain that? It's counterintuitive, no? Do you have some explanation? I don't know really. It's
well, let me tell you that, I mean, we haven't done it, but I think Weaver has published that glioblastoma is way stiffer than normal brain parenchyma. So I think there are many, you know, the fine architecture of these niches within a glioblastoma are far from being detailed and understood. Then if you ask me whether hypoxia can contribute to this, I believe that this is likely also happening. I don't know, simply. But don't be under the assumption that glioblastoma is soft. That's all what I can say. It's not.
Okay. So a lot of question remains open. It's good.
Oh, yeah. Enormous. The field is open for business.
Thank you very much for this fantastic talk. I think that we have to stop now. We will have time for lunch, and we will start again at 2:15 for the last session, chaired by Alain Puisieux. Thank you very much, and bye-bye.
Thank you to all.
Thank you.